The second portion of our review material for this year's AP Biology test is going to consist of material from our second quarter. And even starting out here, I'm confident that this will not be as long as the quarter one video, quite frankly, because we didn't have to cover all that much material in that second quarter. It was challenging stuff, so it took us some time, but there wasn't nearly as much a volume of material. So hopefully this won't be too bad to run through together. And sort of riffing on the light reaction of photosynthesis, we've got the light side and dark side of Star Wars as our theme for this one. All right, so our 10th topic, continuing from last time, is metabolism, right? Photosynthesis and respiration. And so I have the pleasure of telling you this first thing before anything else, and that is that a lot of the step-by-step details that we learned during our Unit 3 are not quite essential for the AP exam. If it hasn't already kind of occurred to you, I demand a lot more of you guys throughout the year than maybe what you really need on that exam. But it's a logical approach, right, in my defense, because one, metabolism I don't think would make as much sense on the whole if you didn't have some of those finer details. But two, if you learn it even more in depth than you need to, that's an advantage in college, but that also is an advantage on this test, because then hopefully when you're faced with metabolism type questions, they feel easy to you. So you don't really need to be able to draw out all of respiration or all of photosynthesis for the test. Certainly this year that's not the case either on the online method. But you do need to know some of the core concepts of these two metabolic processes, which could come into play as the basis of an entire question, or could just be a part of a question. So if we start with metabolism, right, metabolism is all of those chemical reactions in your body, generally dealing with energy, and what you want to remember is that these are often pathways, right? We're going to see this reaction leads to this reaction, leads to that reaction, and so on. Now, respiration, remember, is the major catabolic process, or the major breaking down process of organic biomolecules to get ATP. So if we start with carbs or proteins or lipids, we're going to go through these catabolic pathways to generate ATP from those biomolecules. And don't forget, cellular respiration can occur in all organisms, plant, animal, bacterial, right? If there are cells and those cells are alive, they are probably doing some sort of cellular respiration. Now, the sort of other side of the coin was photosynthesis, right? The primary anabolic process on our planet. And so anabolic meaning building up. This is where we're going to build the biomolecules. So we're going to use that solar energy, energy from the sun, and convert that into chemical energy in the form of primarily carbohydrates. Right, and photosynthesis is primarily performed by plants on the earth, and also sometimes by some bacteria. Now, when it comes to photosynthesis and respiration, and I've included the link here, don't forget one of my favorite little videos, the Oxidated or Love It respiration rat video. There are some things that you definitely do want to know, right? First here with respiration. So the first thing, kind of stemming back from cell signaling again, but don't forget how important phosphorylation is, right? Phosphorylation makes ATP. So essentially what we need to be able to do is take ADP, add a phosphate, and that's our phosphorylation step, right, to get ATP. And so there are different ways that phosphorylation can take place throughout these metabolic pathways. Sort of the main one, I feel like, at least in terms of importance, is oxidative phosphorylation, or oxyphos. Remember, oxidative phosphorylation is coupled to the oxidation of molecules in the electron transport chain. So oxidative phosphorylation is how we make all that a large amount of ATP in the electron transport chain. The second one would be substrate level phosphorylation. And substrate level phosphorylation. And remember, this is not a really scientific way to explain this, but remember that we can make a little bit of ATP in glycolysis and the Krebs cycle. And the way that we do that, or the reason that we do that, is because we're going to take a phosphate away from something, and we need somewhere to put it, right? So when we take a phosphate away from, say, a carb, right, we're going to need to put that phosphate group somewhere, and the logical place is ADP, leading to the production of ATP. And because this involves a phosphate moving from a substrate to ADP, we call it substrate level phosphorylation. And so that happens earlier in the process during glycolysis in the Krebs cycle. Last but not least is stepping over to photosynthesis for a second, and that is photophosphorylation. So remember that we are going to make a little bit of ATP during photosynthesis as well, and that's going to be powered by sunlight, so we call it photophosphorylation. Now the second thing on this slide, or fourth, depending on how you're counting, that's really important here is this term chemiosmosis, or sometimes referred to as chemiosmotic coupling. Now, chemiosmosis, right, is a lot like oxidative phosphorylation, I guess. It's kind of hard to separate the two. But chemiosmosis, or sometimes I prefer the term chemiosmotic coupling, is this idea that oxidative phosphorylation relies on the movement of hydrogen ions across a membrane against their concentration gradient. So remember that when you do oxidative phosphorylation or photophosphorylation, what has to happen first is those electron transport chain molecules have to move hydrogen across the membrane and build up a gradient so that hydrogen can then move back through ATP synthase. Right, so when you see a mention of chemiosmosis, what they're really focused on is sort of this hydrogen movement coupled to, right, the production of ATP. Now, like I said, don't necessarily need the direct step-by-step super knowledge of respiration anymore. However, you do want to have a general idea of what those processes are. And so if you can look back in your notes, we often drew a somewhat more simplified flow chart. A simplified flow chart, I think, would be really helpful for this, because again, maybe you run into just part C on the long free response question refers to something from respiration. You want to have a good idea of what step came before what and what in general happens in each stage. And so that's what we have here. Now, first and foremost here, remember that cellular respiration creates ATP. This is why we do it, right? This is why our cells do this, is to take those large molecules like a carbohydrate and convert it into usable energy in the form of ATP. Now, no matter what kind of organism we are, we're going to do glycolysis first. And remember, word roots here, glycolysis, sugar split. So this is going to split glucose, right, make a little bit of ATP, and generate those electron carriers, NADH. And so again, I don't know if I'm going to have room here, but kind of stage one is glycolysis. Then we've got two options, right? If we have oxygen present, we can do aerobic respiration. If we don't have oxygen available, or we don't have the machinery to do it, then we've got anaerobic respiration. On the aerobic side, we go to the Krebs cycle second, and then the electron transport chain third. Remember, at the Krebs cycle, we're gonna totally get rid of our sugar. We're gonna fully oxidize the glucose into carbon dioxide gas. Also make a little bit of ATP. Again, there's that substrate level phosphorylation, right? And we're going to make more of these electron carriers. And then the electron transport chain is where we make the majority of our ATP, right? And we make that when these electron carriers carry their electrons to the electron transport chain. All right, the electrons pass down the chain of complexes. Chemiosmosis leads to massive ATP production thanks to ATP synthase. And again, remember, chemiosmosis involved that hydrogen movement across the membrane. Now, on the anaerobic side, our second step is fermentation, right? And remember that fermentation does not make ATP directly. All right, so no ATP direct production. Instead, fermentation just recycles the electron carriers. So NADH, it's going to turn it back into NAD+, so it can pick up more electrons again. There's actually going to be a practice problem we're going to look at with this sort of concept built in there. And then we got my fun pictures. Lando Calrissian is one of my favorite Star Wars characters of all time, Billy D. Williams. The classic is still better, but Childish Gambino did a pretty good job of being Lando in the new Solo movie. And then this top picture, I doubt you get at all if you're not a Star Wars nerd. But this is Greedo. Greedo was a bounty hunter that Han Solo shot and killed in the old movies. And there's always a debate of who shot first. You know, did Han Solo shoot him first or whatever? And so over the years, they've actually modified that scene a little bit. So when they released like digitally remastered Star Wars, like back in the late 90s or something, they tweaked the scene a little bit. And then it was a big deal to Star Wars nerds when they released Disney Plus last November. The Disney Plus version of the old movie had the scene edited again. And what they tried to do, at least my understanding, is make it look like Greedo gave Han Solo a reason to shoot him. But what they did is for some strange reason, Greedo appears to say McClunky, and then he gets shot. So who knows what McClunky is, but if you just Google that, you'll find meme after meme on the internet now that just says McClunky, and that's what that is. All right, now just a couple more notes. I forgot I had this slide here, actually, about anaerobic respiration. So we wrote this on the last slide, but fermentation recycles those electron carriers, so glycolysis can happen again and does not directly make any ATP. And then don't forget, and this definitely is something I could see popping up in a question, is that fermentation creates a byproduct, and different cells or different types of fermentation give us different kinds of byproducts. And so in our class, we talked about yeast, which creates alcohol or ethanol as its byproduct. And then in mammals or in your muscle cells, you can sort of kick into anaerobic respiration when you're really exerting your muscles, and you will produce lactic acid. And so where I could see this coming in is maybe there's an experiment or something, and they comment that this leads to lactic acid production, or this leads to alcohol production. And what they're wanting you to pick up on there is, okay, so this is, talking about an anaerobic process, or this cell starting to do anaerobic metabolism. Now, photosynthesis, right? Photosynthesis, on the other hand, creates sugars, right? So it also involves that little bit of ATP production, as we had said, but more importantly, the key goal of photosynthesis is to use solar energy and convert that into chemical energy in the form of sugar. So with photosynthesis, I'd say it's even safer not to really learn details, because as we talked about back in the late fall, a lot of the details of photosynthesis are somewhat hypothetical, right? And so you want to know, the core processes, and you want to know the gist of what happens there, basically. So remember, there are light-dependent reactions, or sometimes just referred to as the light reactions, which require light, right? This is where the energy from the sun is harnessed to fuel a coupled reaction that creates ATP and creates NADPH, right? And then the NADPH and the ATP power the light-independent reactions. Typically, we're talking about the Calvin cycle here, and I think I probably said back in the fall, we used to call these the dark reactions, but not so much anymore, right? Because these do not require darkness, right? These happen 24/7. As long as you have ATP and NADPH to power the light-independent reactions or the Calvin cycle, this can go at all times, and this is where we're going to convert carbon dioxide into sugar. So here is our respiration diagram with some Star Wars ships put on it. But this will be good to know, right? I think if you knew this picture, or maybe, hey, you even have this picture sitting there right by you as to take the test, this could potentially be pretty helpful because it lays out the steps, mentions these different types of phosphorylation, shows you the electron movement. It's a nice summary of those key pieces, right down to electron transport, chemiosmosis, which, don't forget, is tons of ATP production there at the end. Here's a nice simple photosynthesis diagram as well that we used. Also good, right? This points out sort of that cyclical nature of the whole process, where you're going to use these products, ATP and NADPH, of the light reactions to power the Calvin cycle, which is going to create those sugars out of carbon dioxide. Also, don't forget, because it wasn't mentioned on my previous slide, part of these light reactions include using water, basically harvesting electrons from water, and then that's going to generate oxygen, which sort of is a byproduct for plants, which is why they then release it to the atmosphere for us. Now, I'd imagine most of you probably recognize the Millennium Falcon, which is the most famous ship from Star Wars. But does anybody know what this is? Yeah, this ship is called Slave One. Anybody know who it belongs to? Boba Fett. All right, bounty hunter from the old movies. Like you really needed to know that. All right, and then topic 11 is heredity. And although I know it seems strange that this is it, that this is basically the second quarter, right? We spent about half of it talking about metabolism, and then we spent the other half of it talking about heredity or inheritance, right? Sort of that basic level genetic stuff with Gregor Mendel and things. And then it was Christmas. So not a whole lot to worry about here from the second quarter. Now, heredity, recall, does have a lot of vocabulary, right? That's where they're going to get you, if anything. I feel like this is more of a challenge vocabulary-wise when you're faced with multiple choice questions. So that plays in our favor, right? That won't be a case this year. But lots of vocabulary to make sure that we know here. Here's my favorite picture, right, with Gregor Mendel and his very disappointed-looking face with his pea plant and then all the cool other monks hamming it up for the picture. But remember that Mendel looked at pea plants and sort of laid a groundwork for modern genetics. He did not know a lot of the stuff that we now know, and he didn't use a lot of the terminology that we now use, but he's the one responsible for getting things started. And what he looked at are different versions of factors, right, which today we call genes. And these different versions are called alleles, so you definitely want to make sure you're comfortable with that vocab term, allele. Now, some other terms that are sort of non-negotiable, right? You got to know what these terms mean, just in case they do show up. Remember, homozygous means that the individual has two of the same allele. So homozygous dominant could be like big A, while homozygous recessive would be like little a. Heterozygous, on the other hand, means one of each, right? So that would be one dominant, one recessive. And then remember that all of these things are genotypes, right? So genotypes are the actual genes that you have, while phenotypes are the physical expression of those genes. So I believe we already saw a practice problem. A phenotype, remember, is the result of the genes you have plus your environmental exposures. All right, so plus the environment. And these things work together to give you a particular phenotype or physical expression for a trait. Now, Punnett squares can be used to predict outcomes if you have Mendelian traits, right? If you have sort of like simple dominance, as well as a couple other of the non-Mendelian things. But remember that Punnett squares help you predict ratios. I don't think we really have to review how to do Punnett squares. But also remember that Punnett squares are rarely required on the AP test. It's possible that they could be, but it's also always okay to use them if you need to. I could see this for us this year being the kind of thing where maybe you've got some scrap paper sitting nearby, and when you see a question, maybe it'll help you to draw out a little Punnett square on that scrap paper, but then you probably don't have to actually submit that to the AP test. It might just help you answer the question that you have there. Also, don't forget, and we did this back in the quarter one review document, that a lot of times you can compare Punnett squares to actual data using a chi-square test, right? Where your Punnett square is really your null hypothesis, and then you'd be given some actual data for some kind of cross that took place. Now, Mendel had two laws that stood the test of time and continue to be used today when we talk about inheritance, right? Mendel's law of segregation states that the alleles for a particular trait will separate during meiosis. So realize this is only focusing on one trait, right? The alleles for a particular trait. So like the big A and the little a will be separated from each other during meiosis, and the offspring will get one or the other. Right, now, Mendel's law of independent assortment, right, is a little bit different. Right? This states that for multiple traits, the inheritance of one particular trait does not necessarily affect the inheritance of other traits. Now, we learned in class that this isn't always true, right? If genes are really close together, right, we called that their loci, right? If they're on a similar, if they're mapped out close together, their location is close together, then they might be linked, right? So linked genes are an exception to this. But for the most part, Mendel's right in the law of independent assortment that, and the classic example, although it's not really accurate, is like you don't have to have blonde hair to have blue eyes, right? Or in Mendel's case with pea plants, we'll say green peas can be tall or short, right? And that height gene is not affected by the color of pea gene that they have. Now, don't forget that we do have some non-Mendelian inheritance patterns as well, which also, in a couple cases here, can still be Punnett squared. Incomplete dominance is the first idea, right? Incomplete dominance, you might recall the classic example are those snapdragon flowers where you can get red, white, or pink, right? And in this situation, the red might be something like FR, and the white might be something like FW, and then the pink would be, forgot what I was doing there, would be FW. All right, so by saying that these are incompletely dominant, what we're saying is that nothing is really dominant and nothing is really recessive. If you're homozygous for red alleles, you get red plants. If you're homozygous for white alleles, you get white flowers. But if you're a heterozygote, you get a mixture or an intermediate trait, which would be pink. Codominance, also something that you can use a Punnett square for. I remember codominance for our class, we talked about blood type a lot. I did it again. I didn't mean to write that. And for this, remember that we write our alleles with a capital I or a lowercase I, right? So we can have the A allele, we can have the B allele, or we can have the O allele as a lowercase I. And remember, in codominance, what we're saying is more than one thing actually acts as dominant. So that means that you still also have something that is recessive. So with this third allele, this O allele, we see a recessive behavior. The A and the B are both dominant. So you might recall, or you might want to go back and look at that. Remember, type A blood could be two A alleles or could be an A and an O, right? But the only way to get type O blood would be little I, and you also have that weird unique blood type which is AB. And since these are both dominant, they would both be expressed. Right? Last thing that was non-Mendelian that we talked about is polygenic traits. And remember that this is the majority of human characteristics, are things that are polygenic, meaning that you've got more than one gene responsible for the phenotype or for the trait. These you can't do a Punnett square with, right? I mean, you can, but it gets ridiculously complicated. So, again, most of the traits that we would look at in a human involve genes and therefore resulting proteins that are a little bit more complicated than just, big A, little a gives you this. Now, a couple other new vocab terms that we were introduced to during the AP course were pleiotropy and epistasis, right? Pleiotropy, first, this is the idea that most genes have multiple phenotypic effects. So we know today that in genetics, most genes are a little more complex than just this gene gives you this trait. In a lot of cases, a particular gene leads to multiple different phenotypic effects in your body or in your cells. Now, the other one, which is one of my favorite fun examples here, and I included the picture from our original notes with our cute little Labrador puppies, is the idea of epistasis, right? And epistasis tells us that sometimes you have a gene located at one locus or location that actually affects the expression of a different gene, at a different locus, right? And so remember, for these Labradors, they might have this, black or brown trait, right, or locus, but they will also have just a color gene in there. And the color gene will determine whether they express pigmentation or not, right? So if they have, and I'm not real sure what the letters would be, but if they are recessive at this color gene, then that means no pigment, and it really doesn't matter which genes they have in this black versus brown color locus. If they have a no pigment gene, then they will automatically be not colored, right? Whereas if they do happen to, have a coloration trait here, then we have to reference the other locus to figure out if they would be black or brown. Last thing here, which comes into play here in a moment with pedigrees more, is the idea of sex-linked genes versus autosomal genes, right? Remember, autosomal genes are located on chromosomes 1 through 22, so you have a lot more of these, right? But you also have some genes that are expressed on usually the X chromosome, right? But also potentially the Y chromosome, which are your sex-linked genes. And these sex-linked genes, being inherited only on the X or Y, have a more predictable inheritance pattern, right? Males have to get an X from their mom and have to get a Y from their father. And so this can be something that we can observe in inheritance. And also remember that males, we say, are hemizygous for X, meaning they only have one, right? So a male who inherits an X-linked gene always has to express that X-linked gene because the Y does not kind of appropriately compare to the X chromosome. Now we got pedigrees, right? Pedigrees used to predict genotype and outcomes in families. Pedigrees allow us to really determine two things, right? Well, three things. They allow us to figure out genotypes of individuals. They allow us to tell if a trait is dominant or recessive, and they allow us to figure out sometimes if a trait is autosomal or sex-linked. So you want to remember some of these ideas. What we've seen the last few years is occasionally a pedigree-type question where they're going to ask you, and I believe we saw this in our practice problems too, what sort of characteristic would lead a researcher to believe that such and such trait is recessive or whatnot. So what you want to remember, right, most importantly, is that recessive traits are going to show up in a pedigree and they're going to skip generations, while dominant traits are going to be seen in every generation. Right? And then you might be asked, what would lead a researcher to look at a pedigree and think that a trait is sex-linked or X-linked, right? Remember, X-linked traits, again, have to be passed on the X chromosome. So this means that an X-linked trait cannot pass from father to son, because father to son would be the Y chromosome, right? But then most or all of the people shaded with a sex-linked trait will be males because of their hemizygosity, right? But that's going to have to come from mom, right? So the trait would go from mother to son, and then the son would be shaded in. The mom was probably just an unshaded carrier. Remember, the sex-linked traits also are rarely ever seen in females, right? But if they are seen in females, that means both of her X's are affected, and so every one of her sons will have to get one of those X's and also have the trait. So in other words, when we look at a sex-linked trait, sex-linked traits, we're going to see more males, versus autosomal traits, we're going to see more of an equal male-female type ratio. Now again, this could be an actual pedigree that they have you look at, or they might just ask you kind of for a statement of what you would expect to see under these types of situations. And it's usually always pretty safe, if you're looking at a pedigree, to assume autosomal first. Again, there are so many more autosomal genes. That means that there are more likely going to be more questions about autosomal traits than X-linked traits, but you can't rule X-linked out. All right, to wrap things up, I just copied over this slide actually from our notes, looking at pedigrees. And what you want to remember is that you really just need an immediate family usually to tell if something is dominant or recessive. More so, basically, anyone whose parents and kids you know about, you can probably figure out their genotype, right? So again, when we look at this first scenario with widow's peak, when we look at this family, if the parents are shaded in and they have an unshaded kid, right? This means that the parents are definitely heterozygous, right? And the fact that they also have a shaded kid leads us to believe that, okay, this is a dominant trait, right? Whereas if we look at this other picture with attached versus unattached earlobes, there's a huge thing that pops out at us, right? The parents are not shaded in, but they have a shaded in child. What this indicates is that the parents had to be carriers of a recessive trait for it to skip that parental generation. Right? Okay. So I know that wasn't super short, but it was definitely a lot less than what we saw for quarter one. So keep working hard, keep reviewing all of your materials. Again, we've touched on the high points here, but there's probably more in there, particularly in heredity, that you might want to look at, and just to kind of stick it in your brain one more time before you take the test. But keep pushing, keep practicing, and I'll see you guys soon for a review of quarter three and a tiny bit of quarter four that we need to know as well. Thanks for following along.